Hi there, everyone. Hi, this is Mark Cannon. I'm going to talk to you today about dysbiosis and the use of probiotics to correct dysbiosis, and a little bit on the really new and exciting autism research that we're doing. First of all, I have to say there is no conflict of interest to declare. I'm not getting any financial support or sponsorship associated with this presentation whatsoever. And the three key takeaways, well, we're going to talk about dysbiosis and disease origins. We're going to talk about the systemic conditions we see from the dysbiosis and prebiotics and probiotics, but this will concentrate mostly on the probiotics. So this is a probiotic update, the good, the bad, and the ugly, and a lot on autism research. Who am I? Well, I'm a professor in the Division of Dentistry in the Department of Otolaryngology at the Feinberg School of Medicine in Chicago, Illinois. And Feinberg is considered amongst the top of the medical schools in the nation. And they have very strict standards as to what we do for presentations and for declaration of any conflict of interest, which I follow very carefully. Why are we so concerned? Well, it's because the costs of dental disease. If you look at this article from the Journal of Dental Research, going back in 2010, it was almost $500 billion globally. But now in 2020, it's headed toward $1 trillion because of the increase in the middle class in both India and China and the rest of the world. And they want to have good dental care. And we have been providing great dental care, but you would never know it from this article published in The Lancet, where they seem to blame the neglect of global oral health on dentistry itself, saying dentistry needs to be more integrated, in particular with primary care services. We've been trying to do that. The global drive for universal health coverage provides an ideal opportunity for this integration. They want to make us all employees of a big health service, which we know deep down inside that we really care and do the best we can for our patients. And that's why you're here listening to this educational core curriculum. So, can probiotics make a difference? Well, look at this article in Frontiers in Pharmacology. They reviewed all this as the Frontiers in Pharmacology. Would probiotics help to reduce healthcare cost? And they looked at just using the York Health Economics Consortium and the Cochrane reviews at just reducing the incidence and duration of respiratory tract infections, really something very newsworthy at this time because of COVID-19. And they found that just using your plain over-the-counter probiotic could reduce the cost anywhere from $784 million a year to $1.4 billion a year, just if people were to take their probiotics just in the concept of preventing viral respiratory tract infections. Wow. And that's not anything else considered whatsoever, not dental health or anything. And you have the argument that, hey, there's not enough research done on probiotics. Well, let's look at PubMed yourself. Over at this time, 20,672 articles published on probiotics and over 2,500 clinical trials. So people, when they say there's no research, they haven't read the literature at all. There's a lot of research on probiotics, and the vast majority of it is positive, the vast majority of it. So let's look at some dental research now. This was done back when Children's Memorial Hospital was Children's Memorial Hospital. It's now known as Robert Laurie Children's Hospital. So we had a clinical study that was IRB approved to look at what happens to the salivary microbiome after kids use probiotics. And we placed them on two different probiotics, and we found 60 children, 6 to 12 years of age, that were extremely caries prone. Now, don't be misled by us saying 4 carious lesions. They had a lot, but they had to have at least 4 that were big enough that it was well into the dent, because they had to be observable by a lay individual on x-rays, because we had lay people on the IRB, which is the Institutional Review Board. So we collected samples of saliva before and after probiotic use, and they were on the probiotic for 60 days. And then we would do the CRT to measure the level of the caries risk bacteria. And then we do DNA PCR to look at the different genuses of bacteria down to the species level. And then we had two separate statisticians from two different institutions take a look at what would happen after we had placed them on two different probiotics. 30 of them were on Evora, which is from it was from ProBiora, Evora Plus, and Evora Kids. And then we had Perio Balance, which was by Sunstar and was made by BioGaia, Perio Balance. And we looked at the big shift that happened in the level of the lactobacilli and the mutant streptococci. It was very significant. So this is published, and our results were that there was a very significant reduction in the streptococcus mutans and the lactobacilli after the use of our probiotics. And these were the lactobacilli that were associated with caries, not the good lactobacilli. And that was published in the Journal of Clinical Pediatric Dentistry. Then we did a 3-year follow-up because everyone said, yeah, sure, you showed that you can change the salivary microbiome, and it was stable at the time. Is it still changed? And what has been the clinical results of that probiotic intervention that lasted for 60 days? So we looked at the kids 3 years later. We got IRB approval. We wanted to see what happened to them and their caries rate. So we were very fortunate. We were able to bring back, of the 60 kids, where we had all these detailed records where they were on the Perio Balance and the Evora Kids Plus therapy. And we got 53 of those subjects back. Of those 53, only 4 have remained caries active. And they only had, between the 4 of them, 17 additional caries over that 3 years, which was really wonderful results, because you see, prior to the probiotics, they had an extreme caries rate of 5.51 compared to national average of 1.84. And post-probiotic, they had a caries rate of 0.75. So the vast majority of kids became either caries resistant or caries static. So we had a very statistically significant effect on the caries experience of the enrolled subjects. And here's the box and whisker plot showing, hey, look at the difference before and after. Wow, big shift in the caries results. And this is published also in the Journal of Clinical Pediatric Dentistry, titled Retrospective Review of Oral Probiotic Therapy. So again, more statistics. Being IRB approved study, we had to have a number of statistics done by different statisticians at different institutions, with the results blinded, so there'd be no bias. And I was presenting this at one of our international dental research meetings, and a lot of people are very interested in this, going, wow, this actually means that probiotics are far more effective at reducing caries than fluoride. And it is. It's far more effective than fluoride. And you can really take the kids who are still getting caries and put them on probiotics and really have a significant reduction of their caries rate, utilizing, these well-known probiotics. And I went home after that meeting. I found this picture in the basement, and I was like cleaning the basement, from 1975, when I was doing microbiology research on periodontal pathogens, and thinking back, wow, we knew this stuff back in the 70s. We knew that there was pathogens, and we knew that there were commensal bacteria that protected you from the pathogens. Now, there's been validation research. This has been published also. Wonderful publication in Oral Health, and How to Sweden by Savant Twetman, where they looked at immigrant children coming into Sweden, and they took this large group. They reduced the caries rate from 0.8, which was low to begin with, down to 0.2 as a rate. So they had a huge reduction in the caries rate when there was already a low rate to begin with. So that's really a wonderful thing that Dr. Twetman showed us, that it benefits everyone, even the kids with a low caries rate, not just the kids with a high caries rate. So when you're talking about prevention, to have diagnosis, you have to know what is missing, not just what is there, but what is missing. And these are all the things that we actually have used in my office to look at caries risk and so on, from the caries screen, from carie free to CAMX spectra, to the alert 2 oral DNA labs for periodontal disease, mito swabs for mitochondrial health, doing the DNA with Genotech, biome is using the transcriptome, which we'll talk a lot, and just for fun, everyone should do the American Gut Project. The American Gut Project is a very inexpensive way of looking at your own gut and oral microbiome. So here you see mine, and you see if you look at the bar graph, I am under your sample. I'm not your typical American gut. I'm not. I don't have the same microbiome, which I'm really happy about. For the Firmicutes, they are the ones that make you gain a lot of weight. They are over effective with extracellular enzymes, pulling every calorie out of food you eat. I have about half that group that the average American has, which makes it really easier for me to enjoy good food and not have to worry about gaining weight. I think that's wonderful. That's very important. And the other thing, again, just looking at my oral sample here, if you go to the most abundant microbes by the genuses, you'll find I have Veillonella right up there, and third, I have Rothia. Now, the Veillonella are really important in breaking down nitrates, and they make nitrites that become nitric oxide, which is good for your blood pressure, and that's why my blood pressure is low, thank God. And the Rothia are your gluten metabolizers. They break down the gluten in your pizza and your beer. So you can have pizza and beer, and it gets broken down. It doesn't cause you any issues. And as you probably all know, people have sensitivity to gluten. It's because they're short their gluten metabolizing bacteria. They need to restore those gluten metabolizers. And again, if you go down to the different subpopulations and the bar graph, and you see the level of my bacteria are different than the average American. And maybe that's why I haven't had a restoration done now since 1975, when I volunteered to have a fellow dental student complete his boards by restoring a very small caries lesion. But he needed to get through. He had to complete his boards, so it was worth it. This all leads to a lot of the research we're doing in autism, because with these new testing mechanisms we have, like mito swabs, or we can check the mitochondrial health, and biome, we can look at the entire transcriptome. So that's what we've done with 30 patients on my practice who are diagnosed autistic, with a small fraction with my practice, by the way, because we have hundreds and hundreds of children who are autistic in the practice. And then we look at their mitochondrial health, and we'll talk more about this later when we talk about the research in it. And we can look very specifically at each individual species and strain. We can get thousands of different viruses and bacteria and archaea and protozoans and fungus and mold and yeast, all from their RNA, so we know exactly what's going on in their microbiome. So we mentioned autism, autistic spectrum disorder. And what is autism? Well, it refers to a chronic inflammatory systemic disease. Chronic inflammatory systemic disease that produces gastrointestinal and neurological symptoms, and it influences the learning and social behavior. So what is unique about autism is when they've done all these different studies, they know that their gut microbiome is significantly different from kids who are healthy, who have no symptoms of autism. And there's a relationship between the shift in the Clostridia toward Clostridia bolti, histolyticum, and away from Clostridia sporogenous. There's a relationship between that shift and the levels of glyphosate, which is Roundup in the environment. Why things are so different in different communities depends on what they've been using in their farming. So what we wanted to do with this was to determine the difference in the oral microbiome and the mitochondrial health between children in my practice that have autism and healthy children from an area of Colombia, which is designated as a blue zone. Now you need to make sure it actually is a blue zone because many areas that are blue zones aren't. They only seem to have people live longer because people there have falsified their death records to get benefits earlier. If their area of record keeping, like a courthouse or church, was destroyed during a war like World War II, people who were 45 claimed they were 58 so they could go on to government retirement early and no one could prove it otherwise. So that's why people suddenly started to live a lot longer in certain areas of the world. It's a funny story if you read about it. Kind of funny and sad, both. So we looked at these kids ages 6 to 21, and we looked at kids in Colombia age 6 to 16, and we compared everything, their saliva and their mitochondrial health, and there were very significant differences. Very significant differences. And in addition, we took the kids who were autistic in my practice, and for 60 days we had them use xylitol, and then for another 60 days after that, we had them add on the use of probiotics. Very specifically, they added on the ProBiora probiotic. And then we checked at the shifts in their unique taxa, and there was very significant shifts in their taxa. And 6 of the kids who were in the 30 beginning autistic kids group, their behavior changed a lot. There was massive improvements in what they could do and what they could say. They went from being nonverbal to being verbal and small sentences and so on. So in the autistic kids prior to the intervention, that's just some idea. We had 519 unique taxa. Um, in the healthy kids, they had 645 unique taxa, completely different from the autistic kids. After the use of xylitol, they developed another shift where they had 629 unique taxa. So you see the xylitol, when it goes in, it rebalances everything. So you have complete shifts like you can't imagine. We'll show you some research on that because everything shifts a lot in the different types of bacteria. And then post probiotic, there was another shift that occurred. And we had some issues with the probiotic. A number of the kids with autism didn't want to take the probiotic and they had a hard time hiding it. So the results could have been a little bit more dramatic, I think, if we had had total compliance. But this was a transcriptome study, which means we had all the unique functional groups. And these unique functional groups all stand for something. That's the RNA that was pulled out from this high throughput technology all correlates to something in the KEGG orthology function. And that's the Kyoto Encyclopedia of Genes and Genomes. And those numbers stand for something unique that's going on. And we had a lot of changes. For instance, let's look at the KO5046. That stands for gamma aminobutyric acid. That's the chief inhibitory neurotransmitter that is important in the development of the mature mammalian central nervous system. So this is really something important to know about, right? We know how important it is to know the microbiome because going back to just 8, 9, 10 years ago, they started to show in all these studies that bacteria affected anxiety, depression strongly, and your behavior strongly. Study after study after study showed that you could treat anxiety, depression with probiotics, and a lot of them would cause some big improvement in the well-being, mental well-being of the individual, especially ones like lactobacillus formosus and so on, GG, which is a common probiotic that's utilized for many things and big immune booster. And because it affects the level of your gamma aminobutyric acid. So it's, again, affects moods. And then at the same time, they realized there was bacteria that actually were digesting the GABA. They were consumers of the GABA, and that was KLE 1738 bacteria, and that was on the top 10 most wanted bacteria. And so that was something that was very important to discern what that was. And by using 16S ribosomal RNA sequencing, they found the levels of this GABA modulating bacteria the KLE 1738, and they were able to correlate that level to the anxiety depression that was clinically evident, along with it being proven with functional MRI resonance. So they were able to use the imaging and show, yes, this is definitively anxiety depression, which can be determined by functional MRI. And they matched it up to the level of the Bacteroides vulgatus, the Bacteroides fragilis, and also the new bacterium they discovered, KLE 1738, which only grows in association with the Bacteroides fragilis and Bacteroides vulgatus. And fragilis is a very important probiotic that produces your gamma aminobutyric acid, which is a precursor to many of your neurotransmitters. And then Bacteroides vulgatus is associated with autism, associated with periodontal disease, mood disorders, atherosclerosis, diabetes. And it's kind of funny because at certain pHs, Bacteroides vulgatus actually produces GABA. In another pH, it's a major consumer of GABA. So you have to look at the pH, and if something becomes an acidic pathogenic biofilm, you can actually have it go from being a beneficial bacteria to becoming, through metabolic pathways and genes being turned on, epigenetically it can become a pathogen. That's like what happens throughout the entire body. Now, in another KEGG orthology number is the 00780 for serotonin. And that's, of course, very important because, serotonin, hey, what's producing the serotonin? What's breaking down the serotonin? And we do know that there's a good subset of autistic patients that respond to serotonin. In fact, they've actually been studying serotonin first in the lab in vitro, then with mice, a certain knockout mouse that has autistic symptoms. And then now they're doing clinical trials with serotonin for the treatment of autism. Will work in a subset. So we had great statistics. You can look at the nice, pretty charts we have in our Spearman's correlations and so on. We have some great groupings going on, so we have really clean stats, which really surprised us a lot. So let's look at some other studies done looking at the microbiome, just a few of them. Like this is early childhood caries group right here. So they did a full thing again using the transcriptome, looking at this and the importance of carbohydrate metabolic activities. And with this, they showed too, looking at the functional groups, that the early childhood group displayed extensive alterations in metabolic genes, pathways, and modules with enriched functions for sugar metabolism. So when you expose your oral microbiome to high carbs, you shift what is done by the bacteria. And that is fascinating to us because we know that the caries-free individuals had a high abundance of Neisseria lactamica and Streptococcus australis, whereas the high caries individuals had more Streptococcus mutans, which we would expect, but others, Shuttleworthia, who ever heard the genus Shuttleworthia or Osanella as caries, because they were never actually found to be associated with caries before, but now we know that from having the transcriptome. We also know that there's a stable part of the oral microbiome with some very important Neisseria like mucosa and Rothia mucilagiноsa, which we already had discussed how important the Rothia are for breaking down gluten. So this has all happened because in the Neolithic period, 10,000 years ago, when we started to eat grain and we picked up the Rothia, which became very important in the development of mankind. So when you look at the KEGG oral microbiome, you find that's about 20% of these bacteria associations interreact. They're shared by the two different groups, and there's associations that are in between the groups that are like certain species are always associated together, because when you have a biofilm, everyone depends upon the other person. You don't have high growth of strep mutans unless you have Candida albicans, for instance, there. So they need to have each other. They have worked out evolutionary wise to depend upon each other. So if you knock out one, you knock out the other, which is really important. And then you can actually study too the temporal pathways of how sugar is metabolized by looking at these meta-omics, and this is published in the Journal of Society of Microbial Ecology, and that certain positive bacteria like certain lactobacilli and Veillonella, what they do is as it becomes more and more acidic, they help to recover the pH. And one very important one is Streptococcus salivarius, which we know is a very potent probiotic that helps prevent ear infections and sore throats and strep throats, because Strep salivarius inhibits Strep pyogenes, also inhibits Strep mutans. And so what happens as a pH drops, it turns on genes that allow for it to increase significantly the level of ammonia it produces and also iron transport proteins. And so it helps regulate the pH. So our pH in the mouth and the saliva and the plaque is actually regulated by bacteria that are good bacteria. And this is published in scientific reports on dysbiosis and chronic periodontitis. That is not just what we thought, but just, periodontal disease, bone loss, and inflammation, and so on. It turns on genes associated with cancer. 3 genes are turned on that associated with cancer. So it's not just, periodontal disease. When you have this dysbiosis, you get an increase in genetic material that's associated with cancer. They looked at 18 KEGG functional modules in there, and they found your usual pathogens, the red complex. They found some others associated with it. What is very interesting, they found mycoplasma associated with it, viruses and archaea, and a protozoan that is very much so associated with periodontal disease. So you see, there's a whole milieu that you find in this biofilm of everything, which is, why again, when you look at COVID-19 and what happens with the infection of that, because in the secondary infections you get with the bacteria and the cytokine storm. So all this is correlated. So let's go back to results of our research here. Let's look at the KOs that we found. Well, there's a very interesting KO that is 6015 for N-acyl amino acid deacylase. Now there was 37 significantly different ones, and 7 of the 37 are more expressed in the autistic groups for sure for the bacteria. But of the KOs, there's one very, very important one. It was just that KO 6015, which was massively more expressed in the ASD kids than it was in the normal children. So what's going on with that KO 6015? Well, that again, I can mention, is N-acyl amino acid deacylase, and that is a hydrolase enzyme. And that hydrolase enzyme is only from bacteria, and is specifically an enzyme that's produced by Acrinobacter xylosidins. And you're like, why would they have that oral in the kids with autism? It's not that common of an organism whatsoever. The funny thing is, what does that enzyme do that's being produced by that bacterium? Well, it works on your neuromodulators. And that's why they're looking at this as a way to develop new drugs. Ooh, this is interesting. You see, what happens is that the target of that enzyme happens to be NANs, which are N-acyl conjugates of amino acids and neurotransmitters, such as glycine, GABA, and dopamine. That's really interesting. And affects your nervous system, vasculature, and immune system. Now, Probiotic xylosidins, it produces acid from xylose, but not from lactose, maltose, mannitose, sucrose. And it seems to be sensitive to xylitol, which is really important because it shifted in our xylitol patients. You see, you hear about this, and this is in Environmental Science Pollution Research International, because Acinetobacter xylosidins is a new organism strain that likes to colonize high-density polyethylene, and it breaks down high-density polyethylene. So you find it around plastic plants. And, high-density polyethylene is even used in things like baby bottles. But this seems to be very resistant to any antibiotic or antimicrobial. It's a very resistant, resilient microorganism. And like I mentioned before, where do you find it? You find it where you find plastic. So of course, it's far more common in a highly developed industrial nation than it would be in a blue zone nation where they don't use that much plastic. And we use this high density polyethylene, plastic number 2, for milk jugs, apple jugged. We use it for everything. And of course, like I mentioned with our Midas swab data, we're showing significant differences between the group, but we're not quite sure how to interpret it yet. We don't really understand it enough yet to know. So anyway, the transcriptome of individuals diagnosed with autism are really significant from healthy children in the developing world. And these microbiome changes we see may have very, very strong epigenetic consequences. And it looks like we can intervene with xylitol and probiotics to prevent that. And it's not just, of course, autism. All this stuff means we can intervene and prevent things like neuroarterial gingival simplex, which is the systemic disease where periodontal disease is a symptom of the systemic disease. You also get atherosclerosis. You also get Alzheimer's, which we've talked about before. And when do you start that? When you start that early on. You start that when the mom's pregnant with the child. You start that before birth by intervening with probiotics and xylitol. So we do know that there's a very strong association between the gut microbiota and autism spectrum disorder. This is published in Frontiers in Psychiatry. And we do also know that there is something that you can do about it, because the famous study done at the University of Arizona by Dr. James Adams and others, where they went in and they did a microbiota transfer therapy as a therapy to treat children with autism. They went in and they followed up 2 years later, and this is amazing, amazing. Of course, you've heard about this. Everyone's heard about this research. But on the left, under the group A, you see those autistic kids, how their symptomology was from their gastrointestinal tract. These are the gastrointestinal symptoms right here. Severe, they went from so many being severe to minimal or no symptoms 2 years later. And on group B, you see the same thing, but for autism, you see the clinical diagnosis of autism in red, and see how much it goes down and improved by 50%. So 50% of the kids really got much, much better with the microbiota transplant, which is just going back to show what we've shown forever, that normal gut bacteria will restore a lot of the gut-brain communication, bring the brain back to normal. And this has been discussed multiple times, like at the Nobel Conference meeting I attended there in Stockholm, Sweden, and how important it is. And why is it important? Because we do it for our grandkids. And this is my little Henry. It's an old picture of him, but super cute grandson. And I have 2 grandsons, 1 granddaughter. That's why we do what we do. And then I had talked to them when I was there at the Nobel Forum, at the Nobel Assembly, about doing something, some good research with it. And Tor Medved had headed up this study out Karolinska Institute, doing a microbiota transplant intervention on their own in kids with autism. They're very, very surprised to find that there was a good response to it, because after all, as they say, autism is not a brain-based disorder. It's a disorder that affects the brain. It's actually systemic. So lots of positive research being done on that. So I want to thank you all for listening. And again, thank you for attending the American Academy Oral Systemic Health core curriculum.